Hello, my dear students of secondary grade. Today we are going to have a new lesson of studying biology, and we were studying last time the sensitivity in man, and we ended by studying the tissues of the nervous system, and these tissues are composed of nerve cells or neurons, and we are going to complete what we have started last time. Let's start by remembering what we said about the nerve cell. The nerve cell is composed of the cyton and the axon, and surrounded by myelin and Schwann layers, Schwann cells and myelin layers, which are fatty layers. And the cyton has dendrites, and the axon has a terminal arborization, as you see in this picture. We have three types of neurons: the motor neurons, and connector, and the sensory neurons. The motor neurons transmit impulses from the central nervous system to different parts of the body, which are muscles or glands. The sensory neurons transmit the sensory impulses from the receptors or sensory organs, or even the nerve endings spread all over the body from these parts to the central nervous system, which is the brain and the spinal cord. The connector neuron connects between them and locates in certain parts of the nervous system. And now we have another type of cells that compose the nervous tissue, which is called neuroglia or glial cells. And these glial cells, as you see in this picture, locate between the neurons, and they have different functions. Its function is to act as connective tissue that connects between the neurons and support them. They act as insulators, and also they carry out different vital activities to provide the nutrition for the neurons, and also the neuroglia or glial cells have a role in repair the injured parts of the neurons. You know, my dear students, that the neurons do not have centrioles, so they do not divide, and the damaged one is not replaced by another new one. But the neuroglia helps to repair the injured parts or damaged parts of the neurons and to carry out its function. And now the nervous tissue is composed of nerves, and the nerves are composed of many neurons. Let's see the structure of the nerve. The nerve is a group of nerve bundles. Each is surrounded by a connective tissue from outside, which is which is connective tissue, the nerve sheath. And this sheath, as you see in this picture, bounds the nerve from outside. And the nerve is composed of bundles, as we said, and each bundle also is surrounded by sheaths like the nerve as a whole. In between the bundles, there are blood vessels, as you see in this picture, and the bundle itself is composed of many fibers or axons of neurons. So sometimes we call the axons fibers. So when we say the nerve fibers, we mean that it is the nerve axons. And the nerve axon bundle also is surrounded by the sheaths, and each one is myelinated or covered by myelin sheath. And the myelin sheath, my dear students, bear in your mind that the myelin sheath is very important in the transmission of the impulses, because the myelinated axon speeds up or accelerates the transmission of nerve impulse across it, and it may reach 150 meters per second, and the non-myelinated axon or neuron doesn't speed up the transmission. It may reach 12 meters per second only. So it's very essential and very important for the transmission of the impulse. The whole nerve, as you see in the picture, is surrounded by another connective tissue called the epineurium. The epineurium, which contains the blood vessels. Okay, so the axons in the bundles of nerve, and there are some other type of cells that is called glial cells or neuroglia. And now, my dear students, if we need to explain how the nervous system works, I also use some examples for my students to explain how we receive and send stimuli and response to different stimuli during our life activities. For example, if you go back from school and you enter your home and smell the smell of good food that you like, and this smell is a stimulus, is an external stimulus, and being hungry is an internal stimulus. And these stimuli are transmitted into our central nervous system, that is the brain and spinal cord, and they understand what are these stimuli, and they send suitable response to different body parts, actually or in particularly the glands and the muscles. The glands, like the salivary glands, and you feel the secretion of saliva, and the muscles that you speak up and say, I'm hungry, I want to eat, or for your body muscles to move towards the food and start to eat. So if you notice this diagram, you can see these steps. We receive stimuli, internal or external stimuli, and these stimuli are received by the receptors. Some of them are the sensory organs. And these stimuli move along the nerves, the sensory nerves, as sensory impulses until they reach the central nervous system, that is the brain and the spinal cord. And in turn, the central nervous system sends the response as motor impulses go through the motor nerves until they reach the effector organ or the target organ, that may be gland or muscle or both. And now how the nerve impulse transmit along the nerve cell. As we said, that the nerve is composed of axons. So what we are going to study now is the transmission of the nerve impulses across these axons until they reach their target. What is the nerve impulse? The nerve impulse is a message transmitted through the nerve. And the nature of these nerve impulses is an electrical phenomenon. It is an electrical phenomenon with a chemical nature. So we call it electrochemical phenomenon. So the nerve impulses are electrochemical phenomena. And now, my dear students, you need to concentrate very well and to follow the steps of the transmission of the nerve impulses. And the nerve impulse goes across the nerve fiber or the axon of the nerve. So we have this picture and show the structure of the nerve cell. This is the cyton and this is the axon. And by studying the figure, you can notice that there are some positively charged ions outside, and negatively charged ions inside. The positive charges outside the presence of them is due to the presence of sodium ions. And the amount of sodium ions outside is from 10 to 15 times more than their presence inside the nerve cell. So they accumulate outside. But inside the nerve cell, you can notice the presence of potassium ions, which as well they are positive ions. But the amount of potassium ions inside is 30 times than their presence outside. So the greater number of potassium is present inside. Beside the potassium ions inside, there are chlorine ions and protein ions, and these are negative ions. But the negative ions inside is much greater in number than the potassium ions. So the net amount of charges inside is negative charges. And this keeps the plasma membrane is a selective semi-permeable membrane, as you know. And this selective permeability makes the positive ions accumulate outside and negative ions accumulate inside. So the outside of the nerve fiber is positive and the inside is negative. This unequal distribution of ions results in the presence of an electrical potential difference between outside and inside, outside the cell and inside the cell, the neuron. And this equals negative 70 millivolt. So like a pool, we have two pools, positive and negative, and the difference between them equals negative 70 millivolt. The membrane in this case is selective semi-permeable membrane, which is the nature, the normal physiological structure of the cell membrane. It is selective semi-permeable membrane, which keeps some of the ions outside and others inside and prevent the mixture between them. In this case, we say that the membrane is said to be polarized. Polarized means it has two poles. On stimulation, if the membrane is stimulated at a certain point, the arrangement of the molecules of plasma membrane is disturbed. And this results in the disturbance in permeability. So the membrane becomes fully permeable. It is permeable to sodium, which was kept outside. So the sodium ions enters the cell inside, and the negative ions of chlorine and proteins, chlorine also, chlorine only, goes outside. So the charges are reversed. So the inflow of the sodium ions exceeds the outflow of potassium. In the rest stage, at rest, the membrane has what we call the sodium-potassium pump. It keeps sodium outside and potassium inside. But in case of stimulation, it is reversed. So the sodium goes inside, inflows inside, and the potassium is pumped outside, which is not the normal case. So potassium ions go outside through special channels in the membrane. So the channels or the doors are opened now, and the ions are mixed outside and inside. So we notice that the negative charges are kept outside and the positive ones are present inside. In this case, the membrane potential difference equals positive 40 millivolt, and the membrane at this case is said to be depolarized. So it is polarized in normal state, in rest state, but it is depolarized on stimulation or during stimulation. The propagation of the nerve impulse, which is the third step. We said that the membrane at the point of stimulation is disturbed, and it loses its selective permeability. It becomes permeable to ions from inside and outside, and this disturbance is considered as stimulus for the neighboring point. And you can notice in this picture that the disturbance is transmitted into the neighboring point. Okay, so the second point or neighboring point becomes reverse in charges, negative ions outside and positive ions inside. And this is the stimulation, or the next point, or the disturbance that occurs to the next point of the stimulation. The stimulated point acts as a stimulus for the neighboring point, which undergoes the same phases and repeated along the propagation of nerve impulse is repeated along the nerve fiber, and it moves towards the terminal arborization of the neuron. So from the cyton along the axon and ends towards the terminal arborization of the axon. In the same time, during the disturbance of the second point, the first point tries to regain its selective permeability. The membrane regains its selective permeability and becomes impermeable again to sodium. It pumps sodium outside and allows potassium to go inside, and regains its original state. And in this case, when the membrane regains its original state, we say that the membrane is repolarized. So we start with polarization, then depolarization, and the third step is repolarization. It regains its selective permeability again and keeps the positive ions outside and negative ions inside. And this means that it returns to the resting position again. And then some important points that we need to mention in this part. The response of the nerve cell to the stimulus is called action potential. So what happens, that what we have just explained is called action potential, which includes a state of depolarization followed by repolarization. And it ranges between negative 70 millivolt to positive 40 millivolt. That it means 110 millivolt. The nerve impulse is the propagation of the action potential along the nerve cell or the nerve fiber, that is the axon. There is something very important which is called refractory period, and this period is the time elapses between 0.001 to 0.003 seconds, where the cell will not respond to any stimulus, whatever its strength is. And this part, or this time, is the time where it starts when the stimulus starts and when the stimulus ends. So this time is very critical, and the nerve cell does not respond to any stimuli, whatever its strength is. I want to tell you something about the stimulus. The nerve cell or the nerve fiber follows, or the nerve impulse follows the law of all or none. That means nothing, the response is nothing if the stimulus is not sufficient to stimulate the nerve cell or the membrane of the nerve cell. So the stimulus must be strong enough to stimulate the membrane. Otherwise, it doesn't respond at all. Another point that is called the synapse. In this picture, it shows that the nerve cells are connected together or come close to each other at something what we call it synaptic nope or synapse. What is the synapse? The synapse is the functional connection between two neurons. So we have this neuron, which starts by the cyton and then the axon, and the axon has terminal arborization, which come very close to the dendrites of the next neuron. And this is called the synapse. The location is between the terminal arborization of neuron and the dendrites of another neuron. This is the synapse. Now we need to study how the nerve impulse, after it reaches the terminal arborization of one neuron, how it transmits to the neighboring cell or the next neuron. In this picture, you can notice that this is the part of the axon, the last part or the end part of the axon, which ends by a swollen part called synaptic nope or button. So the transmission of impulse ends at the synaptic nope. And this synaptic nope comes or lies very close to the dendrites of the next neuron. So this part locates below it is the dendrite of the next neuron. Again, the structure of the synapse. We have the synaptic nope, and the membrane of this synaptic nope is called presynaptic membrane. And the membrane of the dendrites of the next neuron is called postsynaptic membrane. And the space, the narrow space between them is called synaptic cleft. You can also notice that inside the synaptic nope there are vesicles, and these vesicles are filled with neurotransmitters. The neurotransmitters or the chemical compounds that carry the message through the axon until they reach the synaptic nope. They move in a circular fashion until something disrupts the membrane of each vesicle, and the chemicals flow outside or are expelled outside of the synaptic nope. This is the structure. What about the mechanism of the transmission of impulse? The nerve impulse arrives to the button, as we said, and fill the vesicles. And during the movement of these vesicles, or the presence of these vesicles, makes the calcium ions enter the cell membrane. And these are the calcium ions present in the cleft, in the synaptic cleft. It enters the membrane of the presynaptic nope and presynaptic membrane and ruptures the vesicles membrane. And this releases the chemicals or the neurotransmitters outside of the synaptic nope. I think the picture shows it clearly. So these are the calcium ions that enters the synaptic knob and ruptures the vesicles membrane. So the chemicals are expelled out of the synaptic knob into the synaptic cleft, as you see in this picture. Now the chemicals or the transmitters, these are the noradrenaline and acetylcholine, and these are the chemical or neurotransmitters, noradrenaline and acetylcholine. And these chemicals move across the synaptic cleft or diffuse across the synaptic cleft until they reach the dendrites. At the dendrites membrane, there are receptors. So the receptors of neurotransmitters that are present on the surface of the dendrites membrane, that is the postsynaptic membrane, receive these chemicals. Could you notice them? They receive these chemicals, and then the chemicals transmit inside the dendrite, and so the transmission occurs, the transmission of impulse across the synapse. Once the chemical transmitters bind to special receptors on dendrites membrane, this leads to the change, the permeability of the membrane, and the membrane allows them to enter the dendrites. And the membrane, of course, become permeable to sodium and potassium ions, as we explained before during the transmission of impulse across the fiber. Once the chemicals enter the dendrites, the acetylcholine is destroyed by an enzyme called choline esterase. This choline esterase enzyme destroys the acetylcholine to terminate the action of this chemical. The postsynaptic membrane returns to its restarting position, so it regains its physiological properties and become selective semi-permeable membrane again, and doesn't allow anything to enter it. And now, my dear students, the nervous system. This is the revision of the nervous system structure. We said that the nervous system is composed of central nervous system and peripheral nervous system. The central nervous system includes the brain and the spinal cord and locates at the center of the body. And the peripheral nervous system includes all the nerves come out from the brain, which are called cranial nerves, and the nerves come out from the spinal cord, which are called the spinal nerves. And also there are peripheral nervous system, cranial nerves, autonomic nervous system that are controlling the involuntary actions, as we said last time. Now let's study the central nervous system in details. The central nervous system includes the brain and spinal cord. The first part is the brain. The brain controls the main activities of the body. Its mass is about ranges from 350 to 1,400 grams. That is little bit less than 1.5 kilograms. And of course, the brain controls all the body activities, most of them. It is present in the cranial cranium, and this cranium is a bony sac or bony box, which is the skull. And between the brain and the skull, there are three membranes called meninges. These meninges, as you see in this picture, from outside to inside, they are dura mater, arachnoid, and pia mater. And these membranes are called meninges, and they are very important for protecting the brain and prevent the friction between this delicate tissue, which is the brain, with the bony, hard skull. So it protects it from the friction and absorbs the shocks to avoid the trauma or any damage of the cells. The brain itself is composed of many parts. The first part is the forebrain. The forebrain is the two cerebral hemispheres that occupy most of the volume of the brain, and the two cerebral hemispheres with their cortex are very important for most of the centers that control the body activities. The cortex of the two hemispheres, as you see in this picture, is full of folds and grooves and include the centers for the body activities. The two hemispheres are nearly identical, and in between them there is a deep fissure which doesn't separate them from each other totally, but they are linked internally, and it's a just deep fissure between them. And as we may know, that the right hemisphere controls all the activities in the left side of the body, and the left hemisphere controls all the right parts of the body, and they work in harmony to control all the activities, as we said. Each cerebral hemisphere is composed of many lobes. The frontal lobe, notice this picture, the frontal lobe, the parietal lobe, occipital lobe, and temporal lobe. And this picture is very nice to show different colors to distinguish different parts or different lobes of the brain. There is another fifth lobe, which is hidden by the frontal and parietal lobes. And this picture also shows the presence of the brain inside the skull, and the cortex is full of grooves and folds. Study each part in details. The frontal lobe contains, and here is the frontal lobe, contains the centers of voluntary actions or motor cells, the centers of memory, thinking, reasoning, and speech. All of these centers are located in the front part of the brain hemispheres, and contain the centers control all of these activities. The parietal lobe controls many sensory functions and contains centers of heat, cold, pressure, and touch for sensation of skin. And this locates at nearly the top of the two hemispheres. Yes, and you see the skin centers and motor cells. All of these are centers to control the functions. The third lobe is called occipital lobe, which contains centers for vision, and it locates at the back side of the two hemispheres. Here is the vision centers. The temporal lobe contains centers for smell and speech. This is in green, as you see, at the two sides of the two hemispheres. And one thing I want to tell you, my dear students, that we have one center for writing. If you write with your right hand, so the center of writing is present in the left side of the or the left hemisphere. And if you hand lefted or write with your left hand, the center of writing is present at the right hemisphere. And you cannot oblige yourself to write with both hands unless you practice it for a long time. The second part of the forebrain is the thalamus. Can you notice the thalamus? Yes, the thalamus is here, colored in pink. This is the thalamus, and it controls or coordinates different sensations except smell, different sensations of the body. And the hypothalamus, which is the region below the thalamus, that controls the reflex action and contains centers for hunger, thirst, satiety, and temperature regulation, besides the center of sleep. This is the forebrain. What about the midbrain? The midbrain is the middle part of the brain. It is the smallest part of the brain and represents a connection between the forebrain and the hindbrain. Where is the forebrain? The forebrain is the two hemispheres and the cortex. What's the hindbrain? Is the, yes, the back. So the midbrain is the middle part of it, the middle part of the whole brain. It contains centers for equilibrium and centers related to hearing and vision. And this is locating beside the ears. So it controls these centers for hearing and vision. The hindbrain, it consists of cerebellum. The cerebellum is the back side of the body. This loop, actually it is composed of 3 loops connected by isthmus. And these 3 loops contain furrows and parallel grooves, and is located at the back side of the head. The main function of the cerebellum is to keep the body balance and equilibrium, and it works associated with the inner ear and the muscles. So it's responsible for keeping the body balanced and keep the equilibrium of the body. The hindbrain consists of pons varolai and medulla oblongata, that is colored in light blue. This is the pons varolai and this is the medulla oblongata. And the medulla oblongata and the pons varolai are responsible for the transmission of impulses coming from the spinal cord to the brain and vice versa. Also, the medulla oblongata contains vital centers for respiration, swallowing, vomiting, coughing, sneezing, and blood vessels. So it's very essential for life. If the medulla oblongata is damaged, the person dies immediately. Now, my dear students, we come to the second part of the central nervous system. We studied the brain, and now we are studying the spinal cord. The spinal cord is the extension coming out from the brain, and it runs through the vertebral column into the neural canal. Its length reaches about 45 centimeters long at the back of the body, inside the vertebral column. And unlike the brain, we can find that the spinal cord has the white matter outside and the gray matter is inside, while the brain has the gray matter outside and the white matter is inside. Look at this picture and you can notice the two regions. The H-shaped part in the center of the spinal cord shows the gray matter, and the white matter is present outside. The white matter contains or responsible for the transmission of impulses from different parts of the body to the brain and vice versa, while the gray matter contains the connector neuron and connects between the sensory and motor neurons. Again, the white matter outside and the gray matter inside. The white matter transmits impulses from the different parts of the body to the brain and vice versa. It is covered by meninges like the brain, the three membranes, the dura mater, the arachnoid and pia mater, and these are very essential to keep the brain and spinal cord healthy, and if these three membranes are infected, the person will suffer from severe fever and it may die. From the spinal cord, 31 pairs of nerves emerge to spread all over the body. The right side, like the left side, right and left sides. So they are emerging as pairs of nerves. Now, the most important function of the spinal cord is the reflex action. This is a section of the spinal cord, and as you see, the white matter outside and the gray matter inside, like H-shaped, and the gray matter contains the connector neuron. The spinal cord produces or come out from it pairs of nerves. One goes to the right side of the body and one goes to the left side of the body. And each nerve comes out from the spinal cord has two roots, the dorsal root and the ventral root. The dorsal root contains the neurons that are sensory. So the nerve is sensory nerve, contains sensory neurons. So it transmits impulses from the receptors spread all over the body to the spinal cord. And from the spinal cord there is another root coming out from it, including the motor nerve, and the motor nerve is composed of motor neurons, and these motor neurons go to the target organs that are glands or muscles. The reflex action is the unit of sensitivity in mammals, and when we study how the sensation occurs in the reflex action, we can get all the elements that work together in this sensitivity action. The spinal cord itself has two fissures, the dorsal fissure, which is not deep or superficial, and the ventral fissure, which is deeper, as you see here. Now we studied the structure. What about the mechanism of reflex action? The reflex action starts when we receive stimulus from outside, like heat, and this stimulus is received by the sensory cells, go to the dorsal side and reaches the spinal cord and received by the connector neuron, and the connector neuron sends immediate response like motor impulses to the muscles to pull the hand away from the danger. Reflex action is very important for our life because it makes us avoid dangers, and this is very important for our life. It occurs without any thinking or any time. The time is very, very small, not compared with the responses that we do normally controlled by the brain. So my dear students, by this we come to the end of our lesson today, and I hope you could understand everything that we introduced to you, and I leave you well, and please stay well and study hard for the exam. I wish you all the best, and goodbye.